Okay, now in this session we will talk about the enzyme inhibitors. Enzyme inhibitors are molecules, usually small molecules, that interfere with the catalysis, slowing down or sometimes halting the entire enzymatic reactions. Okay. So, inhibitors of enzymes are basically molecules which modulate the activity in a negative sense. That means, the activity of the enzyme decreases and sometimes the enzyme is totally inhibited, it cannot show any of its catalytic activity. So, these are called inhibitors. Usually they are also very specific, because every enzyme has its own typical active site. The active site has a geometric aspect as well as an electronic aspect, because what is an active site? Earlier the lock and key model which was proposed, it appeared to be a mechanical thing, that the shape of the active site will decide which molecule comes and fits in. But I told you about the induced fit, that it is not perfectly fitted in that sense. What happens here, that it adjusts a little, and there are other interactions. Not only there is geometric complementarity, there are other weak interactions that stabilizes this enzyme substrate complex. These weak interactions are hydrogen bonds, hydrophobic interactions, pi stacking, and the salt bridge, or what are called ionic interactions. Okay. And so, these inhibitors will also be very specific. You cannot have a very diverse array of molecules and that will inhibit a particular enzyme. You should have only few classes of molecules that can inhibit a certain enzyme. Okay. Now, why should we be bothered about these enzyme inhibitors? That is a question, that why do we want to lower the activity of an enzyme? Because many of the diseases, when I have introduced this class, I told that in our body there is a perfect harmony of the receptors and the enzymes working together hand in hand. Okay. And this harmony is sometimes breaks down. What does it mean? That sometimes some enzymes are overreactive. Means if it is overreactive, if its activity is very high, that means the enzyme is produced in large amount than what is required by the body, then what will happen? That enzyme will catalyze a particular reaction, what it is meant to catalyze, and the metabolite, that means the product, will be produced in large amount then. A classic example is, say, many people suffer from high cholesterol. What does it mean? Cholesterol is being synthesized in the body by a series of enzymatic reactions, but somebody is having high cholesterol means his cholesterol level has to be lowered down. So, in that case, what you have to do? You have to now modulate the activity of the enzymes which are involved in cholesterol biosynthesis. This is just a single example, but there are multitude of examples where enzyme inhibitors are actually they are pharmaceutical, they are drugs and they inhibit a particular type of enzyme. It is about 60% of the drugs are all inhibitors of enzymes or receptors. Remember, there is a difference between receptor and enzyme. Receptors and enzymes both have active site, but in receptor there is no reaction that takes place. In enzymes, in case of enzymes, after the binding there is a reaction. In receptor, after the binding there is a signal transduction, signal passes from one place to the other. Okay. Now, let us see that what type of how many types of inhibitions are possible. There are basically two types of inhibitions are possible. One is reversible, another is irreversible. The name suggests that in one case, if you add this enzyme inhibitor along with the substrate, you will without the inhibitor, whatever the rate was obtained, now it will be slowed down. Okay. But reversible means if you can remove the inhibitor again, the enzyme will regain its full activity. Okay. That means the reactivity of the enzyme, which is being perturbed by the molecule, can be again you can again get back the activity, provided you remove the inhibitor. How can you remove the inhibitor? If the inhibitor does not form any covalent bond with the enzyme active site residues, if it does not form any covalent bond, then it will be easy to remove the inhibitor, because the only interactions between the enzyme and the inhibitor are the weak interactions. Of course, this is a collection of weak interactions, but it is possible to remove the inhibitor, because the inhibitor in a reversible case is not bound to the enzyme by a covalent bond. So, that is reversible. And irreversible, on the other hand, this molecule that we are talking about as inhibitor, that goes and binds and then forms a covalent bond with any amino acid residue in the active site. Okay. In that case, it is very difficult to get back the enzyme in the native form, because the inhibitor is already tied up with the enzyme via a covalent bond. So, covalent bond formation or not formation is decides whether it is reversible or irreversible. Now, let us consider the reversible one. Now, again you have usually three classes of reversible enzymes. One is competitive inhibitor. We'll just describe all these: competitive inhibitor, uncompetitive inhibitor, and there is a third which is called either mixed or non-competitive inhibitor. In case of irreversible, again you have two types here. One is called active site directed reversible inhibition, and the other is called suicide or mechanism based inhibition. Okay. So, let us see what are these things. In case of competitive inhibition, remember, competitive inhibition is actually a class of reversible inhibition. So, if it is a reversible inhibition, there is no formation of any covalent bond with the enzyme. So, here the steps are like this, that enzyme, if there is no inhibitor, it will be enzyme plus substrate. Earlier we have seen that you get ES, and then ES breaks down into E plus P. If you add the inhibitor here, which is a competitive inhibitor, so that will also the geometry and electronically that must be matching to some extent to the substrate, so that it also goes and binds to the enzyme. However, in this case they actually are competing for the same active site of the enzyme. Suppose this is the enzyme and this is the active site. So, what happens? In the substrate wants to bind here and then convert it to the product, and at the same time inhibitor also competes to bind at the same active site. Okay. So, enzyme basically has one active site, and this active site is being both these molecules are trying to occupy the active site. It is like that there are two persons, but one chair. So, there will be competition between the two persons, okay, to sit on the chair. So, this is what is competitive inhibition, a competition between the two, and schematically we can show this in this fashion, that you have this enzyme, this is the active site, and this is the substrate on which reaction takes place. So, substrate goes and binds like this, but at the same time there is another molecule which goes also, because this has also got the shape somewhat similar to the active site, and also maybe electronically also it could have some fruitful interactions with the active site. So, this can also at the same time binds here. Now, what is the how the enzyme loses its efficiency? Because some of the active sites now will be occupied by the inhibitor at a particular time point, because both can bind, both are competing with each other to bind, but there is only one available chair to sit. Okay. So, so that actually lowers the concentration of the ES, because at a earlier, if there is no enzyme, if you have two enzyme molecules, both will offer the active site to the substrate. But now what will happen if you have two molecules, suppose one site is now occupied by the inhibitor, another site is occupied by S. So, effectively you are lowering down the concentration of ES. So, that means you are effectively lowering down the rate of the enzyme reaction. Okay. That means the catalysis will be slowed down. This is called competitive inhibition. And the Michaelis-Menten equation takes a different form. We are not going into the mathematics, but earlier you remember in absence of enzyme inhibitor, it was V0 equal to Vmax into S by Km plus S. That was, in this case you have to put a constant, that is alpha, and this alpha depends on the inhibitor concentration, and also it depends on the Ki, that is now called the binding constant of the inhibitor. Like earlier you had Km is gives the binding constant. Actually it is the reversal of the Km gives the dissociation of the enzyme substrate complex, Ki gives the dissociation constant for the enzyme inhibitor complex. Okay. So, that is the equation that it takes. Now, because it is a competitive inhibition, here what happens that if you start increasing, suppose the inhibitor concentration and the substrate concentration are same to start with, but then you start putting lot of substrates into it. So, at some point of time, suppose you have 1000 times excess of the substrate, and the so inhibitor concentration will be very low. So, if you do that, that means most of the active sites at that time will be occupied only by the substrate molecules and not the inhibitor, because the inhibitor will not get any chance to sit into the. That means, now there are chairs, and these chairs will be occupied only by certain will be more. Suppose there are two age group of students, one is say having age 10, but there are 100 boys with age 10, and there is only suppose 2 boys age 15. So, at some point, at any point, particular point, most of the chairs will be occupied by the age group of 10, because they are large in number. So, a similar case happens here, that if you have large amount of substrate, then what happens, that your active sites now will be more accessible to the substrate only. The inhibitor does not get chance to sit on the active site. Okay. Or to put it in other fashion, that the probability of having an inhibitor binding to the active site will be much less if you have large excess of substrate concentration. So, what will happen now? What is the consequence of that? That means, virtually you can remove the inhibition potential of the inhibitor by allowing large amount of the substrate. Okay. Now, what happens? Remember, if you have, say you have started with a particular enzyme concentration, and initially your substrate, you are adding substrate and also inhibitor. Now, what happens, if you want to now have a double reciprocal plot like that, what I said last time, it is also called the Lineweaver-Burke plot. Here 1 by V0 and 1 by S, if there is no inhibitor, that means that alpha, that constant we wrote there, that is 1, then it takes the normal equation. Okay. I can show you the equation again. If alpha is 1, that means it is the original equation, V0 equal to Vmax S by Km plus S. That is the in absence of, that means this is in absence of no inhibitor, that means there is no inhibitor here, absence of inhibitor. And in this case, suppose this is the line. Now, what happens, as you increase the substrate concentration, initially it was your maximum velocity was much less Vmax, because you have inhibitor present. But when you have large amount of substrate, ultimately you will get a line which will be very similar to the line when there was no inhibitor. When there is inhibitor, you will get there is inhibitor in competing amounts like the substrate, then it will be lowered down the Vmax. But when you have large amount of substrate compared to the inhibitor, you will reach the Vmax ultimately for what you used to get in absence of the inhibitor. Okay. And See, that means the value of Vmax, this is important, the value of Vmax remains the same in a competitive inhibition. What changes is, now you need, in presence of inhibitor, you need more concentration of the substrate, more number of substrate molecules to reach the Vmax value. So, the substrate concentration should be more in order to overcome the inhibition, but the value of Vmax remains the same. So, if you have understood this, that means the Vmax does not change, the value of Vmax, again I repeat, the value of Vmax does not change. Now, what if it does not change? Only thing that you need more concentration of the substrate is needed to achieve the Vmax. Okay. To achieve the Vmax. If you need more substrate concentration to achieve the Vmax, that means you need more concentration of substrate to achieve half of Vmax. Again I repeat, if you need more substrate concentration to reach to the Vmax, that means you need more substrate to reach half of the Vmax also. What is half of Vmax? That is, the half of Vmax actually give the concentration which is required to reach half of the maximum velocity is called the Km, the constant, the Michaelis Menten constant. Okay. So, what will happen to Km? So, Km will increase because you need more substrate concentration to reach half of Vmax. Okay. So, that is why, but Vmax value does not change. You remember the double reciprocal plot without inhibitor is this line, and now in a competitive inhibition what happens, as you draw the graph 1 by V0 by 1 by S, you will see that the lines will intersect here, all cut here, because this is the intercept which is 1 by Vmax, their Vmax remains same. So, that means they will all cross at this y axis at the same point. Okay. But what happened to the Km? The Km will increase. Remember, this is minus 1 by Km. So, if Km increases, that means 1 by Km decreases. So, what happens? This is in presence of inhibitor. Now, you have inhibitor, some concentration of inhibitor. Okay. So, your line will slowly go to this side, because again I repeat, because this is reciprocal of Km. So, as Km increases, the value of 1 by Km decreases. Okay. This minus sign may be little bit problematic, but remember, because this is in the minus side of the x axis. So, you can means that we are just talking about the value of Km. So, value of Km will increase, that means 1 by Km will decrease. So, the lines will be like this. So, as you increase the concentration of the inhibitor, you will get further lines like this. So, by doing this Line-Weaver-Burke plot, you can actually determine whether this is a case of competitive inhibition or not. Okay. So, that is all about competitive inhibition. Okay. Now, let us come to this uncompetitive inhibition. This also belongs to reversible inhibition. Okay. Now, here what happens, the enzyme has one active site, always it will have for the substrate, but as the substrate it has got another site, but which was not fitting to the inhibitor to start with. What happens here, when the substrate binds to this enzyme, then this other active site, where which creates a space now for the inhibitor, or I can say which is now more complementary to the structure of the inhibitor. So, now once the substrate is bound, it creates perfect fit for the inhibitor at a particular region, and then that binds. And so, you in this case, unlike the competitive inhibition, here both inhibitor and the substrate are bound to the enzyme. Again I repeat, make it a little bit more clear. So, here what happens, there are two sites to start with, one is for the substrate, but for the inhibitor site it is still not really complementary to the inhibitor. So, inhibitor has to wait, it cannot bind straight away to the enzyme. So, first the substrate goes and binds, and as substrate goes and binds here, then this pocket is perfectly fitting to the inhibitor. So, inhibitor cannot bind until the substrate binds to the enzyme. Okay. So, this is formed, and that is this ESI. So, E plus S gives to ES. Some of the ES will be decomposed to the enzyme and the product, but at the same time some of the inhibitor also can bind to this ES and form this ESI complex. Okay. The problem with this of this binding is that now there is virtually no reaction from this species, where both the inhibitor and the substrate are bound. Now, you can say why there is no reaction, because after the substrate is bound, there is the inhibitor comes and binds, and it is there is some conformational change. I said this is induced fit, nobody gives you a space, perfect space, you have to earn your space in this world. So, very similar, that there is a space, that is true, but when inhibitor binds, there is some conformational change that happens in the enzyme, and that actually perturbs the or prevents the enzyme from doing any reaction on the substrate. So, the bottom line is that ESI does not react. So, what will happen now? That means, your concentration of ES, effective concentration of ES is decreased, because some of the ES, if it binds to I, it goes to ESI, and that does not react. So, this is it's a reversibility here. So, it goes back to ES again and then E plus P. So, this is what is important thing is that I binds, inhibitor binds only after the binding of the substrate, and the second important point is that enzyme substrate inhibitor, these complex, ternary complex, that is catalytically inactive. Okay. Here the equation takes this type of form. Vmax S is the numerator is the same, but earlier it was Km which was multiplied by alpha, in this case Km plus alpha prime into S. Do not be too worried about how this equation has been derived. If you can go to the book by White and White, you will see the derivation of this equation. Okay. Now, the bigger question now is that what will happen to the Vmax and your Km. In case of competitive inhibition, we have seen Vmax does not change, remains the same, but Km increases. That means, there is less binding of the enzyme substrate. So, in this case what happens? The graph is here. So, in this case you will get graphs like this. This is where no inhibitor, this is the line where there is no inhibitor. Okay. And now, as you add the inhibitor, which is working in an uncompetitive fashion, then what you are seeing that your Vmax is changing, because the intercept is changing. Now, in which direction? This is also reciprocal 1 by Vmax. So, that means, this is increasing as you add inhibitor. If 1 by Vmax increases, that means Vmax is decreasing. Vmax is decreasing. Okay. And why Vmax is decreasing? Because the less effective concentration of ES is there, because some of the ES will be bound to that I. Now, what happened to the Km? Km, you see, as you increase the enzyme, the inhibitor concentration, the lines are going towards the left. So, this is 1 by Km to start with, and there is no inhibition there, but as you add the inhibitor, the 1 by Km value is increasing. That means, Km is decreasing. 1 by Km is increasing, that means value of Km is decreasing. So, here Vmax decreases, Km also decreases. Okay. Now, why Km decreases? Just you can say things like that, that if you have, because there are, I said that there are basically two sites, but one restriction is there, that unless the substrate is bound here, the enzyme cannot get itself tied with the other active site. So, here basically what happens, there is induced fit of both the two things together make the complex very stable. So, if the complex is very stable, then dissociation of S is becoming less effective. So, chances of dissociation is reduced. So, chances of dissociation is reduced means your Km is decreasing. That means the enzyme is having substrate sitting in the active site for greater for higher amount of time, okay, because the binding, the association is now higher. Km decreases means your association of the enzyme substrate complex is higher. Okay. So, that is why you get these type of lines. So, if you want that which one, if you have a inhibitor in hand, so first thing that we do, that whether there is any inhibition, and then we try to see whether the inhibition is competitive or it is uncompetitive. And there is one more one which is called mixed inhibition, or that is also known as non-competitive. Actually, non-competitive is a particular type of mixed inhibition. In what happens to mixed inhibition? Here there are two active sites, again two active sites to start with, but unlike the uncompetitive one, here both the active sites are already present. So, the inhibitor now is not restricted that the substrate is bound first and then the inhibitor has to come. That is not, there is no such situation here. Here the steps are like this: E plus S can go to S, I can bind to the S complex, or I can bind with E. It is not dependent on the substrate binding. So, that can go to EI, and then substrate can bind to EI also and make ESI. So, you see the difference. In uncompetitive, substrate binds followed by the inhibitor. In case of the mixed inhibition, the substrate and the inhibitor binding are independent of each other. Okay. But this factor remains, this ESI is catalytically inactive, Okay. Now, what will happen to again that Km and Vmax? So, you will see, because now there is no change in, there is basically no change in concentration, because both the active sites are already present and the binding is independent of each other. So, Km should remain the same, because both are independent, both the bindings are independent of each other. So, Km should remain the same, but your Vmax will decrease, because this is catalytically inactive form. So, some of the ES complex is converted to the ESI, and that is why your Vmax will decrease, and you will see the curve. In this case, both Km and S having this alpha and alpha dash. If alpha is equal to alpha dash, then you have only or that is equal to if alpha is equal to alpha dash, that is what is a case of non-competitive inhibition. So, non-competitive inhibition is a particular case of mixed inhibition, where these two constants alpha and alpha dash are same, and in that case, in non-competitive inhibition where alpha and alpha dash are same, you will now see another type of graph. Here Km remaining the same, as I told you. Why Km remains the same? Because the binding of inhibitor and substrate are independent of each other, but Vmax will decrease. So, these lines, this is the uninhibited one, and slowly as you increase the concentration of the inhibitor, this goes upward, and then that means your Vmax 1 by Vmax is increasing, that means Vmax is decreasing. So, that is the three types of inhibitions for reversible, in reversible cases. We will go to the irreversible cases in the next session. Thank you.